Good. Thanks for the invitation and introduction. Yeah. So today I'm going to present you the work that we're conducting basically during the last 3 years at ESEN, apart from here, and we're interested in host-parasite interaction during spatial dynamics. And to do that, I will start from this kind of cryptic title and from far away, so there and back again. Because there and back again, as you may know, is just the title of the book that Bilbo Baggins wrote when he came back from his adventure in The Hobbit. So basically, Bilbo left home, left the Shire, and through a lot of trouble and adventure, he faced the greed of man and finally arrived to the Lonely Mountain when he faced a dragon, and then basically he came back completely changed. Now, of course, with all the limitation of this image, this is what actually happens with biological invasion and epidemics as well. It's just a parasite or whatever species you can think about. It just starts from somewhere and go through his own adventure, and then he come back completely changed. And let me explain a bit better what I mean with this there and back again, because this is the fil rouge of the talk. And during spatial spread, we have just a parasite we have to catch up and keep the pace with the host to be able to spread and do this life, basically. And during these dynamics, of course, the parasite will likely evolve some traits. And we now know, and we live in that, we know that new variants will emerge. And actually, there will be a feedback on that because this will change also. And here we are with the blue arrow here, back again, this will change the spread of epidemics. So this is what happened. So we have ecology affecting evolution, but then evolution feeding back on ecology. And I'll give you another example of that, a more cartoonish one, to understand what is happening and how this could work. So let's say that I have this fancy name here, spatial epidemiology, which combines metapopulation ecology, epidemiology, and post-parasite evolution. So for some reasons, if you have increase in dispersal, for example, let's say we destroy habitat, and so species has to move around. And so we have our little host, the mannequin, with the yellow squiggle, which is the parasite, that start moving around more, so dispersing more. Well, this would affect epidemiology. And so we have new parasite arriving, changing in infection rate, contact rate in the population, and so here in red, more hosts will be infected. But then if it's a new susceptible host population, what will happen as well is that, well, parasite can go crazy. There's a full susceptible population to infect, and can easily just increase its virulence. It can do a lot of damage to the host, doesn't matter. There will always be another host available to infect. But then this one will feed back again on metapopulation ecology because, well, we will have a crash in demography because we are killing more and less hosts will be available eventually. And so you can see how these things are related and we just feed back on each other. And this is basically what I'll be interested in. And going back to my general and simplified loop, or going from ecology affecting evolution, evolution to ecology, we can ask how does a parasite evolve during an epidemic? But then also we try to close the loop and then how this evolved parasite will affect the epidemic. Now, hopefully we are on the same page and we can start our own parasite adventure. And let's start with the first question also. How does a parasite evolve during epidemics? Okay. There's a lot of things. I show these three circles interacting with each other, seems pretty messy, but can we have some clear prediction or we expect it? The answer is yes, we do have prediction, of course. So here simplified, you have the core in blue with a parasite, again, the yellow squiggle, and I will show you the direction going from the core to the front, so a spatial dynamic with sort of a direction. And what we expect in general is this one. We expect evolution, it's called a dispersal, of high dispersal at the front of the range expansion of the epidemic and spatial dynamic, and low dispersal instead at the core. And why is that? Well, this is also called the Olympic village effect, or it's just a spatial filter. So basically, the individuals that can move very fast and disperse a lot will just run away from the core. So those individuals are the ones that can meet at the front. They will mate, so assortative mating, and the babies, the offspring, will just have the same high dispersal characteristic, of course, if dispersal is a heritable trait. And therefore, literally the front, you just increase dispersal and we run away from the core. The core is just staying there because the individuals do not have those high dispersal characteristics and will just stay in place. So We know this from natural population, from experiment. This works very well. This is the famous cane toad example, so the invasion of the toad in Australia. There's evolution high dispersal at the front. That's what we expect. Now, another key trait, of course, for the parasite is virulence. What we expect for virulence is high virulence at the front and low virulence at the core. And this goes back to what basically I was saying in my cartoonish example with the 3 blocks. If the parasite enters into a new selective population, well, there's the party for the parasite. There's no defense. There's infinite host available. I can just increase virulence. Doesn't matter. You will always have someone else to infect. Contrast to the core, where increasing too much virulence, well, there's the risk of cell shading, so the risk of going extinct because there's no host available. However, this prediction actually holds usually. This is what we expect in general. But there's the thing that these models do not take into account, which is that often parasites rely on the host to go around. So we have parasites, of course, that are not just free propagables dispersing in the air, but also they require the host to go somewhere from one patch to another or to disperse. They require the host to bring them somewhere. And if it's true, well, we can actually have different prediction, which is the opposite. We expect at the front this time low virulence and at the core high virulence. If you're feeling okay, so you have low virulence, well, you can still go around and dancing and go somewhere. It's fine. You can do that. So this is just to say how, if you require the host to go somewhere, this is in the prediction, you require the host to go somewhere. If you have a lot of virulence, so you exploit the host much, but then the host is not going around anymore. So because it's suffering, it's just sitting there. So this is the opposite prediction of the standard. But this is possible. We observe this in some natural population of birds, for example. Okay, now we have some general prediction, remembering just when we consider the host that requires, sorry, the parasite requiring the host to move around. And now we can look for an experimental system where we can apply that. Because, as I said beginning, I'm also an experimental biologist, so just basically do experiments. We do have a system which we found this condition. We have a host, Paramecium caudatum, which is their ciliate. It's just 200 micro, so pretty small, but of course is a microorganism, so has all the advantages. Easy to rear in Falcon tubes, in smaller tubes, in PCR tube as well. Can just reproduce very fast, and also is known to have high dispersal in the sense that it's a very good swimmer. And then we have this parasite, Holospora undulata. This bacteria, pretty big. We can see it in the microscope as well. This yellow squiggle. And as I said before, this is the case of a parasite requiring a host to go somewhere. So these spores can just sit in the medium, so in water, for like maximum 3 days, then they just die. And they can't swim around. They're just floating around the medium, but require the host to go somewhere. Because indeed, Paramecium get infected just filter through the medium. The spores enter the Paramecium and go into the micronucleus, and they just sit there and they start reproducing. And then the spores are released in the medium after host death. So when Paramecium dies, just release the spores in the water. And this is how it looks like under the microscope. Too light, but what is big and purple is just Paramecium, and sort of flames, again, the squiggle is just the parasite. So this is how it looks like. This is like a huge infection. But actually, how we detect that is we take a sample, we put Paramecium on the slide, we have a look under the microscope, and we can actually see the single first spore infecting Paramecium, and we can even, this is destructive, so once we have to throw away the sample afterwards. But still, we can have a look, take samples day by day, and we can see parasite developing and dividing. And this is just at least 2 weeks old infection, which is very big. But we can detect only even one spore because we know where it goes. It goes in the micronucleus. So we can go there, have a look at the micronucleus, and see whether we see the spore. So now we have prediction. We have the model system, and there's another underlying thing which we need, that is there some variation for the trace we're interested in? And the answer is yes. This is how the book does, but doesn't matter. It's just to show you that each column, on the first graph on the left is dispersal, and you have some sort of measurement of resistance and virulence. And each column, each point is a strain. So it's a Paramecium basically. And you can see that there's variability. So these traits can evolve, and we know that it's an experiment. Here in red, you have dispersal evolving over time. You can see that the right one is the right treatment with selective for dispersal. Dispersal is increasing. And we also know from older papers that in this system, also resistance and virulence can evolve. Okay, so we have all the ingredients, prediction, a model system, and actually we know that those traits we are interested in dispersal and virulence can evolve. Now we need to recreate ecological dynamic. So we create epidemics. How we did that? Well, here is a schematic example I'm using, going from core to the front. We just created a system of interconnected tubes. Here's an example. So just two smooth tubes with a corridor in between, which are interconnected. And we can just close and open the corridor when we want. So we started from a single clone. We grow them up, so we have a huge population, and then we infect that population. Once the infection was established, well, we start our long-term evolutionary experiment, where we introduce in the first tube the infected individuals. And then we open the connection. And first, we close the connection. And we decided to maintain sort of a core selection treatment, only the individual that decide to stay in the. And at the front, the opposite was true. So we open the connection for. We allow Paramecium infected and uninfected, right, to disperse. We close the tube, close the connection, sorry, and we propagate only the individual decided to disperse. So we sort of create a core selection treatment, only an individual that decided to stay in place, or the front, only the individual decide to disperse. We transfer this for the core. You can imagine you have much more individuals staying there. So we just take a sample of that to sort of simulate the same bottleneck that the front was experiencing. We propagate those, and after a week, we let them grow for a week, and after a week, we repeated this procedure for basically a year and a half. So we've been selecting host and parasite for a week and a half, sorry, for a year and a half to disperse or not. And afterwards, we start the experiment, when we say, okay, this is enough, should be enough, hopefully. And we isolated host and parasite, and we test for our questions, so what parasite and how the parasite evolved during spatial dynamic, during epidemic, simulated suspension, whatever you can think about it. Okay, let's forget about this for a second. So what we did, we just extract the parasite obtained from these long-term selection lines, 5 and 5 actually. So we have 5 selection lines, 5 replicates for the front and 5 for the core. We extract the parasite, and then we want to know whether it evolved and how it evolved. And to do that, we put the parasite on a host, right? So a host that never faced the parasite before, just to have the parasite effect of evolving into 2 different spatial regimes. Okay. And so we did some parasite adaptation assay. We measure quite a bunch of traits, but of course, 2 fundamental traits we've got clear prediction of these 2, so dispersal and virulence. So as I said, we infected this naive host, and then we measure in similar fashion dispersal. So we reinfect with the evolved parasite naive population. We put in the first tube, we open connection, 3 hours, we close connection. And then we sample and we have a look at how many infected individuals disperse from one tube to another. So the proportion, so dispersal was the proportion of infected individuals moving from the introduced tube to the second tube. So this is how the propensity now of the evolved parasite to disperse or not. And then we measure virulence. So from this population, we isolate single individuals, and we just put 1 single Paramecium in a PCR chip. You can see there. And then we follow how much they grow and when they die. So the 2 measurements of virulence. Now, the extra thing we did, which is very interesting thing of this study, is that we also had an extra measurement, an extra technique. So we basically built an epidemiological model where we fit we fit some population dynamics. So we use this epidemiological model to fit and having independent estimates of parasite parameters, which means that we just built a simple SI model, so coupled differential equation, knowing a bit how the system works. And then we have here some just, as you can see, it's just sample of density. So taking a sample from the water, counting how many Paramecium you have in infected and uninfected population. And this was fed to the model. And we obtain, of course, extract some parameters like virulence, transmission, etc. So this is just this is nothing to do, this is an extra thing. And we use it to obtain independent estimate of the key parameters that we expect for the parasite important. So transmission, virulence, infectiousness, and other things. And to see whether our data, our experiments, we measure, so dispersal, virulence that I just explained, match with some model as well. Okay, let's have a look at some results. So how about parasite dispersal? So on the Y here you have what I said, the proportion of individual dispersing. Again, color codes are always the same. Blue is the core parasite, and each point here is an evolutionary replicate. And you can see more time 1 point because of what we have technical replicates of that. And what is the bar is not box plot. It's just model prediction. So we run the model and we plot the prediction on top of our data. And the line is just the average model prediction. The weird panel on top right is just to confirm basically our stats. So it's just the difference of the model prediction of core minus front. So if those predictions are the same, there's no difference, of course, and you will observe a distribution there on the top right panel, this peak overlapping with 0. Because if they're the same, it's just a number minus the same number equals 0. Okay. What you observe here is that the front parasite is now allowing the host to disperse more. This is highly significant. It's actually twice the level of the core. So parasite that evolve at the front is now allowing the host to disperse more. And this is basically what we expect. Low evolution or low dispersal at the core and evolution of host dispersal, high host dispersal at the front. For virulence, here is the result. Again, color code, each point is an evolution line, and the point are model prediction. These are uninfected individuals, so our own, coming from those treatment. So no parasite was present. You can see there's basically no differences. Indeed, the panel on the top right is just overlapping with 0. And the first panel on the top is the maximum number of individuals produced in a tube. So basically the peak of their growth. And at the bottom is how many replicates survive. So no differences for the front. However, when we look at infected individuals, so what we're interested in, you can clearly see that the red one is above blue, confirmed also by our simple subtraction method. And this means basically that the front produce higher number of individuals and more replicates survive, meaning lower virulence. So basically the parasite is affecting less the host this time. And this one was confirmed by our model prediction. So this is a posterior of our epidemiological fit. And you can see how what the model says, just think, okay, this is just a population dynamic of my system. So just the density of time. And how about virulence? And well, it gives exactly the same thing. Low virulence at the front and high virulence at the core. And this is again what we predicted. And basically what we interpret this is that there's a prudent exploitation of the parasite at the front. So the parasite at the front is now low virulence, and likely this allows the host to disperse more, which is kind of what we predicted from other papers. Now, if you make brute force of these two traits together, so just plotting here a giant mean and standard error, this is where the core parasite sits, certain level of dispersal and high virulence, and front parasite is just moving there. So it's increasing dispersal and reducing virulence. So we interpret this as an emergence of a dispersal virulence trade-off due to the fact that the parasite is required to follow the host, and so only the parasite that are less virulent actually allow the host to disperse. And so parasite is affecting less the host dispersal. And okay, this is nice and all, but do we have an idea of the mechanism that could be behind this? Because I just say, okay, this evolved, but why this is happening? And what we did as well is we look at infectiousness. which is the development of parasites over time. So every day we went there and sample and have a look, as I said at the beginning, at the microscope, and have a look how many spores we see, what the parasite was doing. And what happens is that basically the front parasite it takes longer to develop. So to go from first infection to high infection load. Compared to the core parasite, you can see here again, this is just model prediction. The core parasite has a steeper and therefore a faster production of spores. So the core parasite requires less time to produce spores. And this indeed was confirmed again in our epidemiological model. You can see a peak 140, meaning less time, and while for the front parasite it takes longer to produce those spores. So if you put all the information together in a multi-path analysis, you have sort of an explanation of what's going on. So there's a reduction in infectiousness. So the front parasite, it just takes longer to develop. Therefore, it suffers less and is able to divide more. So reduction in virulence, and therefore allow the host to disperse more, so it can go around easily. Okay. So with this, we have first idea of how does a parasite evolve during an epidemic. We see adaptation of the parasite of the necessity to going around the hosts, and we see the emergence of a dispersal virulence trade-off. So something important maybe to integrate when we think about epidemics or dispersal dynamic is to know how the parasite disperse and what they do to their hosts. Okay, now before to go to the second part of the loop, so the there and back again, so I'd like to open just a little parenthesis on the host to be able to add another point on this consideration. So if you remember, this is what we did, and I say, well, we isolated the parasite and we tested on the host. This is just what I showed you. But then what we did as well is that in this population, there's epidemics going on, so during these tubes, but there's also uninfected hosts around. So what we did, we isolated a evolved host that was uninfected, asking how actually those dynamics could also affect host evolution and not just parasite. Plus, we have, of course, in addition to these two treatments, other two treatments where parasite was absent. So population, we always went for the same spatial treatment, so they were allowed to disperse or not. No, sorry, allowed to disperse and then we propagate those to disperse or not, but without a parasite. And basically, we have 4 treatments. We have a host that evolved without a parasite, a host that evolved in population with the parasite, going through course selection treatment, and the same for the front with individual dispersing but without the parasite in the population and individual dispersing with the parasite in the population. From this, say I just isolated hosts that were uninfected, just to see the host. And we then there measure a bunch of traits, dispersal, some demographic traits, so growth and capacity, and also swimming behavior, so how the hosts move. Plus, we measure resistance, and in similar fashion, we use naive parasites, so parasites that were in the stock culture that never faced these hosts before, and we measure resistance. And this is actually how the video looks like. So we take a sample and we look at how they move. We have a script that is able to localize the individual's Paramecium, and then it's telling us how they move, how fast they move, how much they turn, their size, and how many individuals we have. Okay, some results from host trait evolution. Okay, this is super messy, but it doesn't matter. I don't want to go through it. I'm going to focus on the top 2 panels, which the first one is dispersal and the second one is resistance. So the first 2 columns are the hosts that evolve without a parasite, and the second 2 of the first panel, talking about dispersal, is dispersal of individuals that face the parasite in their evolutionary history, but they're not infected. Okay, they're just being evolved with the parasite. What we observe here is that dispersal is going down. So basically, evolving with a parasite reduces your dispersal ability in general. And when we infect this population with a parasite, what we observe is actually, yeah, they decrease dispersal, but they're now more resistance, which kind of intuitive. You face a parasite in your evolutionary history, you become more resistant to the parasite, compared to population that were going through some spatial regime, but without the parasite, so while investing on resistance. Now, if we put together all the other traits, what we have, and what we did is multivariate analysis. And the thing I would just want to say here is that the full points are the infected population, empty points the uninfected. And there's a just separation clearly between evolving with the parasite or without the parasite. So this is just to say that the host will evolve some dispersal syndrome going around. So you have many bunch of traits. There's a lot of interaction going on in these traits. And however, the parasite will just modify those and will move to a different multivariate space. So basically, expanding and or not with the parasite will just modify your whole phenotype. And this is the point I just wanted to add here. The parasite evolved during an epidemic, yes, and adapt to likely to the necessity of dispersing and keep track with the host. There's this dispersal virulence threat of emerging, but also the parasite affect host evolution, moving them to different trait space. Okay, now time to go back and try to close the loop. How evolution will feedback and affect the epidemic dynamic. So second part of this derivative again. And to do that, well, we lost, because this is lab experiment, we lost our evolved parasite. And so we had to start over again. So we basically repeated our front and core regime again for an extra year. And we start front and core parasite just to test how these now are able to change and modify epidemics. And this time, instead of going there and using this parasite to measure some parasite traits, what we did, well, we went to the next level and we built linear landscape. This is how it looks like in the lab. So a system really of interconnected tubes, not just two-patch system, but several tubes connected in order to simulate epidemics. So we have evolved parasite, we extract that, and we infect naive population again, and we put it in the first tube. And then we went there and we slowly open collection and keep track of when the parasite was arriving, how many infected individuals there were around, the density, how fast they were moving, and things like that. Basically, this is indeed back again. So we want to know how evolved parasite would affect the epidemic spread and also the speed of an epidemic. Now, we can't really do with all the patches because of the limits of our old school technique of taking a sample looking at the microscope. But what we did is we look at what was going on in patch 1, 3, and 5. So asking when the parasite was arriving and once arrived, what was doing basically. Okay, this is how epidemics look like. Okay, for some reason, there should be a lot of points behind, of course, but there's no appearing reasons. This is therefore this is just the means of our measurement. What you can see that overall, already you have front parasite producing high infection prevalence over time. So on the y you have infection prevalence, the proportion of infected individual in the population, and over the time that we measured in patch 1, 3, and 5. You can see that the front seems to have high infection prevalence overall. Now, let's ignore for a moment patch 1 and focus on 3, because this is our spatial component. We want to know how the evolved parasite front affect the speed of the epidemic, so when the parasite arrived in patch 3 and 5, and also once it arrived, what kind of epidemic it produced. All right, so let's take a look at 3 and 5. And what we see here, this time we have proportion of parasite that arrive in patch 3 over time, and the same for 5. Now for 3, what you can see is that, well, current front, tend to arrive at the same time point. But for the front, we have high proportion, high replicates arriving on that patch. And when we look at 5, this is even more evident. It's clear that front parasite arrives 2 time points before than the blue in patch 5. So it takes 8 time points from the parasite to reach patch 5. It takes 10 time points, and this difference is like basically 5 days. Is a lot for the core to arrive at the same patch. So it seems that the front parasite is a better spreader. And once it's there, what the parasite is doing, well, this time we have just log of number of individuals per ml in the patch. And you can see clearly difference in slope. So the front parasite has a steeper slope, and so produce epidemic peak much faster than the core for either patch 3 or 4. Now, how can we explain that? Because here I'm just showing, okay, this is what we observe. Again, can we explain that? What is driving this? Okay, it seems that the front is faster and produce epidemic peak faster as well. But why? What's going on? Well, what we did is that we have to do, and again, some evolution assay to understand what's going on. And what we did is we measured transmissibility. Again, here, for some reason, there's a lot of points on the back. That does not appear here, but doesn't matter. The front parasite tends to be more transmissible than the core. So basically, we took 20 infected individuals, we put it in a PCR tube with some other 100 individuals, and after the first day, we went back, we sampled all the tube, and we checked every individual to see how many were infected, because we can detect with our technique how many new infections we have. We know that that one is an infection, that's a non-infection. What we obtain is that the front parasite is much more transmissible than the core, although it doesn't seem so, but if you see the distribution on the back, so how many points we have, you will see like a really different distribution of transmissibility. So this one in itself would explain our pattern, because if there's more transmissibility, well, it means that we have more infected individual in the population, and just by that, it's more likely that some individual will just swim and go infected, go on the other side, just because there's more individual around that carry the parasite. That could be already a very simple and basic explanation of what we observe. Plus, there's another thing to add, which is again we measure virulence. This time, this is a decay function of number of individuals over time. And you can see how the front curve just decays smoother, meaning that this is, of course, less virulent, as we kind of, remembering, we obtained in the other evolutionary experiment. So a repeatable result, of course. And we can add virulence on top of transmissibility. Basically, if the transmissibility is maybe potentially more likely to disperse more, and also if they suffer less from the parasite, well, this could create some sort of differential dispersal, because the individual that had the front parasite red, well, they suffer again less from the parasite, and they're more likely to go around and swim to the other side. So this is really work in progress, and because we get our hands on final data like a month ago or so, maybe less. So we don't jump to the conclusion, but still what we observe is that the front parasite is better spreader and produce faster epidemic peak. And this could be done just by two simple traits here, transmissibility and reduced virulence. Now, question is, how is the relative contribution of transmissibility and virulence? That's the big question. And so we have now two PhD students that are gonna help us to understand what goes on, building an ecological model where they're just gonna try to understand the relative contribution of transmissibility and virulence in explaining our pattern, even if just changing transmissibility would explain what we observe. Okay. So to sum up, this is what we've been doing. We went from there, asking how ecology will affect the evolution of parasite, and we observed a little twist also hosted, and how parasite evolution affects epidemics. Well, still work in progress, but it seems that there's clearly an acceleration of epidemic when the parasite evolved. And this is difficult to observe. So just to stress out how we use also with a combined approach, and we're going to do it for the back again. We need to integrate density, dispersal, stochasticity of the system to understand what's going on. It's just not an experiment. Our exercise of epidemiological fit, and also the next model, which is coming up, is going to be super important to understand what's going on, because as I said at the beginning, you have these three blocks: the metapopulation ecology, the epidemiology, and evolution occurring at the same time. So just doing experiments is hardly enough to understand what's going on. The good point is that in our simple system of interconnected tubes, we can replicate epidemics, and we can observe these dynamics. And the last thing I want to say is that we're going to do a last effort, last huge experiment, and we try to disentangle what's the relative contribution of host and parasite in affecting epidemic spread. Here I just show how parasites affect epidemic, but as we know now, even the host evolved during this time. And so we have evolved host, evolved parasite, evolved host without the parasite. So with a huge effort coming up soon, we'll be able to test and cross all these lines and see and disentangle what's the effect of the host on the epidemic spread, what's the effect of the parasite on epidemic spread, how much evolution is necessary to explain epidemic pattern that we have. Okay, and with that, I would like to thank all the collaborators that worked with me on this project, the institution, my fund, the Swiss funding, now the INF funding, the Australian collaborators, and you for your attention. Thank you very much. I have to have questions.
So, thank you, Giacomo. So, if you have any question in the amphitheater or even in the via Zoom, you can ask your question with the text via Zoom and normally in the room. So let's start. Don't hesitate.
Yeah. First of all, thank you for your talk. I thought the experiments were like really elegant and very nicely with your models.
Thank you.
I have a question because I don't know anything about this system. If there's any role of like co-infection that could be, if you imagine the individual that gets co-infected with like the core strain and the front strain,
yeah,
that you get this kind of competition because one is developing more quickly. And if you think that would like what that might do to this kind of simplified model system.
Yeah, I never thought about that because we can't do that. You can imagine, okay, I infect with core, a mix of core and front, and finally you have differences, and I don't know why. But actually, I don't know. Because it seems that one is more infectious, one transmits less, and one transmits more, like the front, but it takes longer to develop. So
yeah,
I don't know.
And that's negative feedback.
Yeah. I haven't thought about that just because I can't produce that. It was super interesting. Like competition as always. And you can just do it equally. Things that you can track and here. Yeah. But yeah, intuitively, there would be a lot of feedback going on, but front parasite winning, I would say, because less virulent. And once they are infected, like we have a huge belly, they do not get infected. They can't get co-infection anymore. There's a little narrow window at the beginning when you introduce the parasite. Because we also have other parasites in the lab that do kind of similar stuff. So they can get co-infected. But it's just this narrow window of a few hours after you introduce the parasite to the population.
So it's almost like an establishment.
Yeah.
Thank you.
See.
I have also it's a curiosity question. Like, what is the mechanism of resistance to the parasite, and does it has a cost for dispersal or growing?
Yeah. So the mechanism is unknown. Sadly, have some ideas, of course. One. So there's the Japanese and the Russians and some Germans group for reasons, historical reason, that are interested in that. And what it seems likely is that there's two possibilities. One is they basically just behavioral avoidance. they just stop filter feeding. They feel sick individual around, which is possible that they're super plastic as these individuals. So they start eating less, for example. Or there's a very strong mechanism of defense. So when they filter feed, Paramecium is known to have a lot of vacuoles. So we just go into these acid vacuoles and they just kill the spores. What happens, this happens all the time, but then the infection arises when the parasite is able to go out of the vacuole and migrate to the nucleus. So mechanism is the Paramecium for some reason is super good at avoiding parasite escaping. And for the cost of resistance, that's what I kind of show you quickly, which is the parenthesis on the host. It seems to have a potential trade-off with dispersal, and there's a tendency as well for growth to be reduced, as you expect. But it's not super clear, it's not that neat, so it's just tendency. And you can see things going on just when you look at all the traits combined together. You really see, okay, this is how population infected or not separate, and this is how they separate. And yeah, that's what we observe.
I have one question, I'm not sure. Because you look at the individuals who disperse, but then when you disperse, you also have some bottleneck effect. And I was wondering if maybe because there's less, I mean, is the density of the role in the evolution of the parasites? I mean, if there's any possibility to see it for the density is always the same for the core population or the front population, or we did the same, or
we adjust that indeed for sort of control for bottleneck effect. So of course, for the front, we just propagate the entire population dispersed, which is a small fraction, like 10-20% max. And for the core, well, just to take a representative sample, 20%, so we produce the same bottleneck. That's what we do. But yeah, this is a thing for sure, and this is the way we control this by doing it. So creating a similar bottleneck. But it's nice how I get always this kind of question from people working on population dynamics or genetics.
yeah,
Yes.
for the
Thinking about
genetic-
gene surfing or other kind of things.
And so for the genetic part, because I'm going to genomes,
Yeah.
so I mean, do you have any people looking at the genome of the parasites that are in the point that want to see if there's any? I mean, where are the differences?
Yeah, we don't know. Coming up project. Yeah, you don't. Has I think. Well, yes. Starting collaborating with guys from Germany and Italy as well. Are interested in the sequence. What's going on? So of course we have those frozen parasites somewhere. And now a matter of sequence and see whether we can have some genome resolution for what's going on. Yeah.
You have a question in the chat.
Yes. Okay, I'll just read out loud. Hi, can you please clarify the assumption of density in your spatial model? Is the core less dense than the front and vice versa or opposite? I'm wondering about how selection will play out in competition. Yeah, good point. On my spatial model, no, there's no different assumption for host density. And the core is actually, so as I was explaining here in the audience, we standardized that. So there's no, there were no like huge differences or no different over time for density, because we control for that. And what we see, however, is that of course the parasite evolve and affect as a feature population density core and front now. But that we explain that as an emergent property of the system as we control for this during the long-term experiment. Hope I replied to the question.
last question, maybe in the chat? No? Okay, in the room. So thank you everyone to this seminar, and see you on Monday, exceptionally, because we have the AG on Thursday. So have a nice day and bon appétit, because it's time for lunch.
Merci. Thanks everyone. Bye.